The CellSeal Cryo Vial offers flexibility in use compared to other cryostorage containers. To fill the vial, use a standard luer lock syringe, draw a small volume of air into the barrel of the syringe, and aspirate the desired volume of fluid to fill. To fill the vial, connect the luer lock to the fitting at the top of the fill tube. Ensure the connection is solid. If resistance is felt during fill, ensure that the connection is completely closed by twisting an additional quarter turn. Slowly depress the plunger to fill the vial. Continue depressing through the extra air to evacuate the fill tube. The Clear Access CellSeal vial allows filling using a syringe or a pipette tip. Completely depress the pipette into the open fill tube and slowly dispense the fluid. Because the tube will be open, gravity will evacuate the fill tube. To prepare vials for cryogenic storage, seal the tubes using CellSeal RF Sealer. Place one tube between the jaws of the sealer and depress. Hold the jaws closed for an additional 3 seconds after the light extinguishes. This will ensure a solid weld and container closure integrity. To retrieve materials from the vial, cut or nick the vent tube distal to the microbial barrier. Remove the foil debris cover from the bottom of the vial. We recommend sanitizing with IPA before accessing the contents. To puncture the septum, use a non-coring technique or non-coring needle. Position the bevel of the needle away from the septum and press against the septum. Gently aspirate the material from the vial. By placing the lumen of the needle at the interior surface of the conical base, you will ensure complete collection of your sample.